It's noon, so I think we're going to go ahead and get started. This is Nicole Kleinstroyer. I'm here with Cynthia Ryder and Warren Casey, who's going to be giving the first half of our webinar leading up to the Converging on Cancer workshop that will be Monday and Tuesday of next week. So the first talk is going to be given by Dr. Warren Casey, who's the director of the National Toxicology Program's Interagency Center for the Evaluation of Alternative Toxicological Methods. And he's also the sponsor for one of the new Health Effects Innovation initiatives under the leadership of Brian Barrett, the Associate Director for the NTP. We have multiple new HEIs, and one of them is focused on carcinogenicity. So, Warren, take it away.
Okay, thanks very much. And yeah, so I want to give a little bit of background on NTP and these new HEIs to help provide some context for what's going to happen in the world of carcinogenicity testing. First, a very high-level look at, there are really two NTPs that people refer to. One is the big NTP, which is a multi-agency partnership, and that is headed up by Linda Birnbaum, who's also head of NIEHS. And then there's what's referred to as the division of the NTP, which is the group located here at RTP. Brian Barrett was appointed the director as of January 2018. He took over for John Bucher. And one of the things Brian is hoping to do is to kind of bring a new approach to what has traditionally been a kind of standard guideline study program in many respects, and to kind of expand the influence of the Tox 21 approaches across the entire program. So we actually have a new vision and mission. The vision is to improve public health through the development of data and knowledge that is translatable, predictive, and timely. And we're really focused on that timely aspect, especially for cancer testing. A two-year bioassay here at NTP typically took, from start to report, 8 years or more. And if we're dealing with something that's really a threat to public health, almost a decade is really too long to get that answer. So we're really focused on ways to get human-relevant information in a much faster manner. And then the mission of how we're going to do this, we really want to address public health issues through collaborations with stakeholders and global partners. We recognize that this is too big to be done alone, and the only way to do it is through partnerships and collaborations. We are focused on environmental hazards of public interest, and I'll get more to the legal aspect of that in just a minute. And we want to actually lead the transformation of toxicology through the development and application of innovative tools and strategies. So typically at NTP, we've had several different types of programs. Probably the single agent program is the one most people are familiar with, where we take one chemical at a time and do an assessment on that. But we also look at agent classes such as PFAS, PAHs, and herbals. We've also done a few quick react kind of programs, such as Elk River or Deepwater Horizon, when we really need to get information to the public as fast as possible. And so what we're doing now is what are termed health effect innovation areas. And there are three of them: developmental neurotox, cardiovascular, and carcinogenicity. And the goal is to move towards a model where the deep understanding of human pathobiology informs the evaluation of hazard and risk. So there's really going to be an increased emphasis on the human relevance of what we're doing. We recognize that one of the big challenges we face is this continuum of pathobiology. Unfortunately, we work within regulatory systems, and even we're forced to communicate to the public in ways that put chemicals in boxes or classifications. Something is toxic, it's not toxic, where we all know it's a lot more complicated than that. So we want to focus on understanding this transition between normal and the adverse outcomes. So just as an example, with the estrogen receptor pathway to breast cancer, so this is an example of an adverse outcome pathway, which identifies a molecular initiating event and then follows that through mechanistically to some type of adverse outcome. Now, the thing about AOPs is they describe a process that we know about. They do not describe all of the processes related to this that we do not know about. So just because something starts up at the top with the molecular initiating event does not mean you're going to have an adverse outcome. It's very probabilistic. There could be 30 other pathways incorporated here that aren't represented that terminate before an adverse outcome happens. So that incorporation of the probabilistic nature of toxicology is something we have not historically done a great job at. So we'd like to understand the mechanistic relevance of these pathways and be able to identify that transition area where we don't have to wait for an adverse outcome to happen, but where we have a high degree of confidence that it would happen if it continued. So this is something that Brian came up with. It's called the Translational Toxicology Pipeline. It describes how we at NTP will be taking on every project. So this is not just related to cancer. And it's supposed to represent a more deliberate approach to answering specific questions. So instead of getting a chemical in and doing a standard guideline study to answer a typical question, we will always start at the base, developing knowledge to see what in silico computational tools are around, identifying in vitro studies that could be relevant, and then moving up the animal studies from short-term to long-term. So we're not just going to jump in with the chronic in vivo studies. And the other aspect here is the continuous integration of knowledge. And again, I think that's something that as toxicologists we really have not done a great job of, is learning from every single study we do and incorporating that learning back into the process. So as Mark Twain said, the secret of getting ahead is getting started. We've been talking about this for almost a year now, and it's time to get going. Last week, we had our first organizational meetings for all of the HEIs. This is the program management team for carcinogenesis. The folks were selected based on their diversity of expertise in either cancer or specific fields that are very relevant to what we'll be doing with the HEI. So when it comes to what does NTP have to do, what are we congressionally mandated to do, I went back and looked at the actual language in the Act. And it's interesting in that we are charged to list all substances that are known to be human carcinogens or may reasonably be anticipated to be human carcinogens and which have a significant number of U.S. residents exposed. We're also charged with informing the information concerning the nature of those exposures and the estimated number of persons exposed. So most people tend to think that NTP is all about hazard, but it's very clear that exposure is a big part of what we need to start considering, or we need to consider going forward. To date, we have run about 600 two-year bioassays, and out of that work and other work put together with the data, 248 substances have been identified, 62 known to be human carcinogens, and 186 reasonably anticipated. And the reason I've highlighted the substances there is it's important to keep in mind that we don't just do chemicals. It can be the effects of shift work, it could be the effects of viruses, crumb rubber. It can be a wide variety of things other than just chemicals. I think the other thing that strikes me here is that we've done 600 bioassays, and about 40% of those have some association with cancer. That's obviously not, we're obviously testing chemicals which have some kind of suspicion This isn't just kind of a random 40% of all chemicals are associated with cancer. So we're obviously looking at something that's given us reason to think these were carcinogens. And I think building on that type of knowledge in the future is really what we need to focus on. It seems like a lot of these studies were essentially done to understand the doses that would cause cancer and maybe some more specific mechanisms. But it's obvious that there's plenty of information out there that indicate chemicals could be of concern. This is the typical process used by NTP: epidemiology, animal studies, mechanistic studies, and then the use of newer data. That last column, we haven't done a lot with when it comes to cancer assessment. It's something we need to get better at. But the important thing here is that we are not obligated to use any specific approach for carcinogenicity testing. So there is no policy or guidance or guideline that we have to follow to determine whether or not something is carcinogenic. And that's a huge advantage compared to some of our colleagues in other agencies, which are very much bound to policy and specific guidance. This is going to allow us to work with those agencies and develop approaches that they probably couldn't do. And then by us working the paradigm, and hopefully we can have some parallel testing, this is one way that they will eventually be able to adopt an entirely new approach to risk assessment for carcinogenicity. We're certainly going to increase, we're going to keep doing what we've been doing, but we're going to increase the emphasis on human relevance. The role of in silico and computational approaches is really going to be at the forefront. If you look at what's being done with some of these technologies now, it's clear that they offer a huge advantage over a lot of even the in vitro approaches. Comparative physiology is another thing we really have not done a very good job at in the past, but we're really going to need to take advantage of all the data that's been generated in those animal studies to try to help inform human risk. And then mixtures is something that there's no doubt that the testing of the future has got to be able to figure out how to incorporate knowledge of mixtures. So this is just an example of a paper that came out in Cell last week, which I think represents a great approach that we will certainly be looking into. Again, it's using human relevant information to identify signatures of specific classes of carcinogens. And this is just an example. There are many, many papers coming out now that touch on this type of approach. We also have to take more of a holistic look at what we're doing. It can't just be focusing on trying to replace a specific animal test. And this article in Nature that refers to a whole journal, a whole issue of the American Statistician is a good example of that, where we have got to move away from the reliance on t-tests and p-values to determine what's significant or not. We really need to focus on what's biologically relevant, and there are many, many approaches that can be used. And there are so many studies, really good studies, that have been discounted simply because the p-value didn't reach a certain predetermined threshold. And I think we really end up throwing away a lot of valuable information if we rely on outdated statistical approaches. So one of the things we will certainly be doing is following this strategic roadmap that was developed by ICFAM, which is the Interagency Coordinating Committee on the Validation of Alternative Methods. So this is a group of 16 agencies that work together for almost 3 years to try and determine why there was such a poor incorporation of new technology into regulatory testing. The conversion of the knowledge that we're generating, the new approaches into regulatory testing has just been really bad, to be honest. It's for whatever reason, we're just not making that transition at the pace that we need to. So this project was really geared towards finding out why that's the case and developing an approach to remedy it. And what we found was that there really needs to be a circularization of the technology process. Historically, it's been very bottom-up, where technology is invented, then they try to validate it, then they hope somebody's going to use it. But it really has to be a completely integrated process. And most important, it has to start with the utilization. Technologies that try to figure out what regulators want, guess at what they want, or guess at what industry needs, have historically done very poorly when it comes to being adopted. So if we can get the regulators, the end users, and industry involved at the very first step to help guide the development of the technology, that turns out to be absolutely essential. It's not just the technology, it can be a new approach like we're using with the cancer HEI. So we recognize that the very first step we need to do is problem formulation, and we will spend many months doing that. And we also recognize that there are other efforts, many other efforts that are relevant to what we're doing. So the very first thing we're going to be doing is a scoping exercise to understand how we can best fit into all this other work that's happening across the world. And finally, I think we also need to consider communication. It's something that, as scientists, we think we're pretty good communicators. We publish, but we tend to miss a larger audience. That's the public and Congress, especially when it comes to things like cancer. It's going to be very important for us to be able to clearly convey what we're doing when it comes to assessing these chemicals, because we do tend to put things in buckets, carcinogens, not carcinogens, it will cause cancer, it won't. And we need to do a much better job of conveying the true nature of what's going on and helping people understand the concepts of risk and relative risk and that type of thing. It's something that we haven't done a great job of in the past, but we're going to make a concerted effort to do it with this particular HEI. And that's all I have. Thanks for allowing me to give this webinar, and we look forward to working with you all starting at the workshop.
Thank you very much, Warren. I forgot to mention at the beginning, if there are questions, if you could please type them in the chat window, and we will address them that way. And we're going to be sort of forcibly muting people's lines just to make sure that we don't get too much background noise. So we have about 10 minutes for questions. If we want to take any from those of you on the line, please go ahead and put them into the chat window. And I can see. So we have a question on the availability of the slides and whether or not they will be. And so they will be posted hopefully by the end of this week. We're working towards that. If not, then next week. We do have another question that just came in. Are cumulative exposures being considered?
Yes, I think we're going to have to consider cumulative exposures. I don't know how we're going to do it, but that's one of those things that I don't know how you get around if you're doing a realistic assessment of carcinogenicity.
So in addition to the 3 health effect innovations that Warren mentioned, we also have different focus areas, and one of them includes combined exposures and mixtures. And this whole project that we'll be talking about at the workshop is kind of the intersection of those 2 things, so definitely.
Yeah, and I didn't list, when I listed those 4 areas, those weren't all the areas that we're going to have programs in. As Cynthia just mentioned, mixtures is a big one. Capability development, there are a number that will be published once they are finally determined.
Okay, we have several more that came in just to me. If you want me to read them aloud, that's fine. So what process will NTP use to engage across the many stakeholder communities they represented in the logos?
So we are already involved at different levels with those organizations on different projects. I would imagine that we will try to summarize all of those interactions in some type of white paper or some kind of publicly available document. Almost everything, well, everything we do will go through some kind of public commenting period, either at our Board of Scientific Counselors meetings or at some other venue, like a workshop. So once we pull all those together, we will articulate all of what we found in some type of document.
Okay. Another question is there any alternative to repeated dose toxicity being discussed in the consortium?
Yes, I think I had that on a slide. I may have deleted it just for need of space, but repeated dose, especially in the context of how we do in vitro or in silico to simulate repeated dose exposures, is going to be another critical aspect of this, just like the mixtures is.
Okay, we have one comment, just happy to see an emerging discussion about statistical significance versus biological significance. Another question, how is the concept of risk calculation addressed for identified carcinogens present in regulated products on the market?
I don't have a good answer for that right now. Yeah, you know, as I said, we just started this last week, and we're still trying to develop the problem formulation and the scope of what we'll be working on. So hopefully as that emerges, we will have discussions and workshops and opportunities for people to ask those questions and for us to provide better answers. Nicole?
Well, and I think that goes back to the first question about how we're engaging across our many stakeholders, because the concept of risk calculation is something that, you know, regulators deal with on a day-to-day basis. You know, we don't set exposure limits or make regulatory decisions, but we do provide the science that underlies the basis for those regulatory decisions. And so making sure that regulatory decision makers and risk assessors are involved in those conversations from the very beginning is critical towards being able to implement some of these new approaches in that context.
Yeah, and that is one of the things that the roadmap addresses, is that the need to not only understand the regulations needed by the various agencies, but also their data needs and their context of use. And that's something that we've already started working on, because you can't, it's not right to say the FDA needs something. CEDAR needs something completely different than CIFSAN. Different divisions within EPA have different data needs. The Department of Defense has their needs. So the very first thing we're going to do is try to understand all those and what impact we could have by making certain assessments on what they're doing. It's going to be really important for us to understand the implications of everything we're doing.
And fundamentally, what are the questions that are driving those data needs and those decision contexts in terms of human health protection?
Okay, the next question, could you say something about how NTP is contemplating integrating the results of its testing effort into the evaluations by OHAT and the report on carcinogens?
Well, they are going to be part of this. So we have a representative for the report on carcinogens. We're still looking at how NTP is organized and what that new structure is going to look like. So I'm not sure how to answer that, given that things may change going forward with the organization. So, but the people involved will, in those current roles, will play an active part in what we're doing with this HEI.
Okay, the next question, concerning the use of new tools by regulators, it is important to understand what is needed to establish that connection, what is needed for regulatory decision-making. That might just be a comment.
Yep.
Okay. Next question, how do you deal with general talks based on in silico tools?
I'm not sure I understand the question. How do we deal with general talks?
General talks based on in silico tools.
Maybe instead of focusing on a specific question about a specific toxicity, how do you deal with a complete unknown?
Yeah, and that's something we really haven't gotten great at. And that's why, animal models will continue to be a very important part of what we do going forward. I don't know if probably the two-year bioassay, if we have a decision tree, will be part of that, but I imagine there are a lot of other animal models that will be very helpful going forward because the complexity of mammalian physiology is something that we are not even close to modeling yet.
Okay, the next question, how do you foresee two-year rat bioassays being used in the future?
Well, as I just said, I think they would probably be a last resort. I don't think it's advisable for us to try to say we're going to replace the two-year bioassay. I think that would be something that's very hard to do, but I think we can start using mechanistic data in a way that makes the two-year bioassay less relevant and less relied on in the future.
Okay, we're still good on time?
Mm-hmm.
Okay. Yep. Still rolling. How do you evaluate whether one change is normal, adaptive, or maladaptive?
I don't know. That's something that that's really going to be the whole focus of what we're trying to do.
Mm-hmm. Okay. Next question, will there be any opportunities for extramural research? I think she continued, possibly call for filling data gaps.
Yes, and again, that's something that we address in the roadmap, the need to identify sources of funding and data gaps and try to get those linked together. I think certainly NIH, as the largest funder of biomedical research in the world, will play a big role in this, and hopefully we'll have some influence on the areas that really need to be looked at, especially in terms of human risk assessment.
And the last question that I have is, how do you plan to engage the environmental NGO community in this effort?
I think we'll engage them just as we do with all the other partners. We're open to participation and collaboration with everyone from PETA to HESI to CropLife and all the federal partners. So there will be opportunities to reach out or to be engaged with us through workshops or other projects. And we will always publicize those in the Federal Register whenever there's an opportunity for that. Or just email me and get in touch.
And just to add on to that, specific to environmental NGOs, this workshop, what part of the genesis of this workshop, if you saw Cynthia's talk last week, was some interactions with the Environmental Working Group and a nomination that they submitted to the NTP based on the outcomes of the Halifax project and the hypothesis that was generated from that work. And so they've been very closely involved in the planning of this workshop as well.
Okay, we got one more question that came in. How does NTP NIEHS plan to integrate the results of its testing effort into the hazard evaluations that the NIEHS conducts currently at OHAT and ROC?
So, yeah, that gets back to the earlier question. I think
it might be just repeated.
Certainly, OHAT is, the approaches that OHAT uses are going to be critical to this, the systematic review process. I imagine there would be some harmonization of systematic review processes used, and then we're bound by law to come up with the report on carcinogens. So that's going to be a mainstay of what we do. I imagine a lot of the information that they use will come out of this and also, in turn, rely on those two groups to generate.
Okay, great. Thank you so much, Warren. So now we're going to turn it over to Mark Fielden, who's giving the second talk in this webinar hour. So Mark is the Scientific Director at Amgen, where he's been for the last 10 years. He currently leads a group of toxicologists and an investigative toxicology lab and has a long interest in developing new methods to assess carcinogenic risk of pharmaceuticals. He is the lead of a subgroup that I'm also a part of, working with the key characteristics of carcinogens and trying to integrate that framework with the hallmarks of cancer and what's understood about cancer biology, and then trying to develop a set or identify a set of assays and biomarkers to effectively measure both the key characteristics and the hallmarks as it applies to environmental carcinogenesis. So hopefully we can get control transferred over.
So Mark, you currently have control of where it's going in presenter view. If you go up to display settings in the top left, you can click swap, I think is what it's called, and then it'll take it out of presenter view. I have you muted. Apologize, you were muted. Yeah, you should be able to share and then click display settings and then click swap after you share.
It looks like I'm sharing. Do you not see it?
Yeah, sorry. Once you hit start the slideshow, I apologize.
Okay, hitting slideshow mode. Bit of a delay.
Okay, and then display settings at the top left, besides show taskbar.
Oh, I see. I've got two monitors. I'm looking at the wrong one. Gotcha.
And then swap presenter view and slideshow.
Perfect.
Great. And then, yeah, the Skype for business. Yep, good to go.
Yep. Okay, great. Well, thanks for the invitation and opportunity to present. I feel like somewhat of an outsider here. I represent myself. I've got no conflicts of interest. I'm trying to change my slide. I'm not sure if it's going.
We are not seeing in advance, no.
Either am I.
Usually just clicking or hitting like the right arrow should make it advance. There we go.
Okay, great. So just a disclaimer, the views expressed here are my own, and I don't have any conflicts of interest. Like I was saying, I feel like a bit of an outsider to this group working in pharmaceutical industry, but I think we all have a common goal of improving cancer risk assessment, whether it's compounds in your environment or chemicals in your medicine cabinet. So I appreciate this opportunity, and it's been a rewarding experience collaborating with the folks at NTP and others. Right, so I think this group is familiar with the key characteristics of carcinogens, but just to make sure that everybody's on the same page, I'll at least define what I mean by this, spend a little time talking about how it integrates with the hallmarks of cancer, but then spend most of the time talking about results of a working group that I've been involved with, led by Martin Smith, involving folks at NTP like Nicole and Cynthia Ryder and others, focused on recommended assays, both in vitro and in vivo, for measuring the KCs. So the key characteristics of carcinogens were originally published by Martin Smith and colleagues back in 2016 in Environmental Health Perspectives. And it represents a set of properties of known carcinogens. And these properties are believed to contribute to their carcinogenic effects. And the way they came up with this list was by studying a list of known human carcinogens, basically the IR class 1 compounds. By evaluating the mechanisms of these carcinogens, they were able to identify a number, quite a large number, of mechanisms that were thought to contribute to their carcinogenicity. For pragmatic reasons, they distilled that list down to a set of 10 characteristics, and these became the 10 KCs, as I'll refer to it in short. And I think people in this field will readily appreciate a lot of these mechanisms of carcinogenicity, such as is genotoxic, induces chronic inflammation, alters cell proliferation, cell death, etc. Now, as one would look at this, I think you could easily draw some parallels with the hallmarks of cancer, which was a well-known, well-cited publication back in 2000 by Hanahan and Weinberg. And this publication outlined a number of characteristics of cancer, and that's the key difference here. So the key characteristics of cancer describe the properties of carcinogens, whereas the hallmarks of cancer describe the properties of a tumor and the mechanisms by which they can evolve. You can see the list of hallmarks on the right here. The original hallmarks were published back in 2000, and then subsequently in 2011, an additional secondary paper came out that expanded upon the original hallmarks, integrating a lot of new information that's been learned over the years. The other point I wanted to make is that most tumors, if not all tumors, exhibit multiple hallmarks at various stages of the tumorigenic process. So that's the hallmarks, and by direct comparison, these are the key characteristics. Again, properties of carcinogens rather than properties of cancers. Similar to hallmarks, many carcinogens induce multiple KCs. And just by looking at the list of KCs on the left, you can see a lot of parallels with the hallmarks. And even though the wording might be similar, there's not necessarily a direct a one-to-one relationship. So this working group discussed extensively how the KCs relate to and can be integrated with the hallmarks of cancer. And there was a lot of interesting discussions. But at the end of the day, I think we agreed that there really wasn't a clear one-to-one relationship. Cancer is a little bit more complex than that. So in this diagram, what I'm trying to represent here is that carcinogens can impact a number of KCs, one or more KCs at different points in the process, be it initiation and or promotion. And these characteristics facilitate or give rise to cell transformation, and that transformed cell, that ultimately that tumor, will exhibit one or more hallmarks. And again, these hallmarks can occur at various points in the carcinogenic process. Not only that, carcinogens can impact not only the formation of new cancers, but can promote existing cancers. So I'm thinking about KC10, for instance, and the induction of angiogenesis, which would have the property of facilitating growth of pre-existing cancers. Another point about the KCs is that they often have converging effects on the genome. So there are a number of KCs that ultimately give rise to DNA damage, albeit through different mechanisms. So KCs 1 and 2, for instance, you can think about DNA adducts being caused by an electrophilic compound that can give rise to mutations. Other genotoxic events that can directly give rise to DNA damage. And then there's a related KC, KC3, alters DNA repair or induces genomic instability. Quite different forms of DNA damage, but nonetheless alters the genome. And then there's yet another set of KCs that can give rise to DNA damage indirectly through alterations in oxidative stress, oxidative DNA damage, which when repaired or misrepaired can give rise to mutations, all of which can initiate or augment a number of hallmarks, for instance, mutation of tumor suppressor genes or oncogenes, for instance. Now, you might be asking, well, how can we use these KCs to improve cancer risk assessment? So in the original paper by Martin Smith back in 2016 in EHP, they described how they utilize these KCs to organize a large body of mechanistic literature to help understand the carcinogenic risk of molecules. In this particular example on the left, they're illustrating how a large body of literature with benzene, for instance, can be evaluated and separated and categorized into the component KCs, such that each individual KC could then be further evaluated for the weight of evidence and the specific mechanistic data that might support or refute the involvement of that mechanism of action. Now, since then, there's been a number of efforts to try to quantitatively integrate the KCs, put some quality metrics around the data to add a little bit more objectivity to that systematic review of compounds. Now that's, so this is still very much a work in progress, a systematic review. And I just give one example here with Danielle Wyckoff and colleagues from Tox Strategies, but there's other approaches out there. And this works well for evaluating those compounds for which a large body of literature exists, such as benzene, but doesn't necessarily provide a solution for those compounds that have yet to be tested. So another application of the KCs is to use it as a framework for developing a toolbox, which would allow one to assess new compounds. So in this scenario, along the top, you've got the 10 key characteristics. And what we're envisioning here is a set of assays, both in vitro and in vivo, that one could utilize to assess a novel chemical, a novel drug, for instance. So in this process, a panel of in vitro assays could be used as a hazard identification screen. Now, we wouldn't necessarily prescribe a checkbox approach whereby every chemical would be evaluated for every KC. Depending on the compound, depending on the application, one would have to choose the appropriate set of assays. So a tier 1 screen might be hazard identification in vitro, but as we can all appreciate, what is observed in vitro might not always translate in vivo. So what is needed here is a set of assays or biomarkers that can be evaluated either in animals or in humans to assess the degree by which in vitro hazards might translate in vivo, and importantly, to assess the exposure response characteristics, which would be important for human risk assessment. So for this toolbox to be useful, it should account for a broad set of potential carcinogens, not just the ones we're used to today. Unfortunately, my PowerPoint wants to reload. Just bear with me for a second here.
We can always
Sorry.
run your slides from here, Mark, if you want, or, oh, it seems like it reloaded pretty quickly. That's the fastest time from failure to success I've ever seen a computer achieve. Can you hit the display settings again and swap presenter and slideshow?
Yeah, it's dealing with two monitors.
Unless that will cause it to fail again.
Yeah, it's losing my mouse on the... I'm suggesting you quickly revert to plan B. Doesn't want to cooperate, and I hesitate to watch you guys watch me fumble around. I don't trust it.
No problem. We will take control back and display your slides, and then you can just let us know when to advance.
All right, I think it was on slide 10.
Second.
It was bound to happen.
Okay, just bear with us, everybody. We'll have them up and running in just a second. Okay, slide 10. Slide 10, yep.
All right, I'll just give you the word for the next slide.
Okay. Next. There we go. Okay.
10. Right. Okay, good. So I was saying that for a toolbox to be useful, we should consider developing a toolbox that would be useful not just for the carcinogens that we know of today, but for potential carcinogens or compounds, agents that we might want to evaluate in the future that we perhaps haven't invented yet. I'm thinking about novel drug targets, new modalities, coming from the pharmaceutical industry. I'm thinking about gene therapies and other gene editing approaches like CRISPR-Cas9. There's probably a whole host of other chemicals that you guys are thinking of. It's worth pointing out that the original list of KCs were developed based on studying these IARC Group 1 carcinogens, which is a finite list of well-established human carcinogens that were, for the most part, mutagens, although it was quite diverse. There was viruses in there, irritants, UV radiation, for instance. But hopefully a toolbox would be broadly applicable to future agents that we might want to evaluate as well. So I think that needs to be considered when developing the assays and whether or not the 10 key characteristics are broadly applicable to all potential agents. Okay, next slide. So with these challenges in mind, a working group was established, and this was really spearheaded by Martin Smith, together with Kate Guyton from IARC, as well as Nicole Kleinstruer. And the goal of this working group was to outline or recommend a number of well-established in vitro and in vivo assays or biomarkers to assess the KCs. Now, in going through this process, it was obvious that there was some gaps in our toolbox, and so part of the process here is outlining these gaps and as well as highlighting some emerging approaches that we think warrant some further investment. So the net objective of this approach is really to facilitate research in the advancement of new tools to aid in cancer hazard identification and assessment. So it's really a starting point, I think. So next slide. So I'll kind of walk you through some of our thinking and some of the work that we've been doing. We're still in the process of collating some of the work into the form of a paper. As we initiated this process, we identified this paper here, written back in 2016, which attempts to do a similar, attempts to go through a similar exercise. However, this is really focused on a selection of functional assays to evaluate the hallmarks of cancer. So you can see the parallels here. But again, because the hallmarks of cancer represent properties of tumor cells and not necessarily carcinogens, there are some key differences. But in the terms of the assays that we find useful, there is going to be some natural overlaps. So I just wanted to highlight that. Next slide. So rather than go through an exhaustive list of assays, I just wanted to highlight a couple just to illustrate some points and make some clarifications and kind of walk you through some of our thought process. So KC number 2, so this KC is called Is Genotoxic, and as I indicated before, a number of KCs impact DNA damage. So I wanted to kind of illustrate our thinking around this approach. And it was really a pragmatic one. So for this specific KC, we restricted the endpoints to both mutations as well as chromosomal alterations, specifically classigenicity and aneugenicity. So these are really the genotoxic endpoints that are informed by what I think of as some of the classical gene tox assays that we routinely utilize. The standard ICH S2 battery for assessing pharmaceuticals, for instance, make use of these tools. Some of these tools are restricted to in vitro evaluation only, like the Ames assay, but others like chromosome aberrations and micronucleus can be evaluated both in vitro and in vivo, including humans. And then we also added some additional considerations for some emerging assays. So the TGX CDI is a transcript-based signature for identifying genotoxic molecules. So this is an emerging approach that was recently published, but it has been worked on for quite a number of years. So the approach here wasn't to be exhaustive and to list every single assay out there. As you can imagine in the gene tox field, There are dozens and dozens and dozens of GeneDux assays that have been published, but it was really just a recommended list, and I think of it as more of an illustrative list of assays to illustrate the types of endpoints that one would assess for each of the KCs. Next slide. So by contrast, KC number 3 is focused on DNA repair and genomic instability, very distinct endpoints from what is measured in KC number 2. And again, some of these assays are still emerging. Next-gen sequencing, for instance, I think has a lot of promise for evaluating some of these larger genetic events, such as duplications, amplifications, and chromosomal rearrangements. Next slide. So in going through this process, we identified a number of gaps in the toolbox. So for instance, KC number 6 induces chronic inflammation. There's just not a well-established in vitro assay. You can imagine it's very difficult to model chronic effects acutely in a Petri dish. It's classically evaluated histopathologically in vivo assays. Another major concern is whether or not these assays are really appropriate and linked to a carcinogenic process, or are they more correlative? What's the degree of validation behind these assays? And are they all relevant to the carcinogenic process? I think there's a lot of questions around whether or not some of these KCs are specific. Oxidative stress is one example that's been discussed. So I think only once we have sort of a defined list of assays and some agreement around the extent of validation can one start rigorously or systematically addressing some of these questions about their relevance. Not only that, how do we integrate the results of multiple KCs? I think that's an active area of research. Next slide. So I do want to highlight a couple of KCs since they are emerging in terms of some of the assays that have come online, but do represent a significant gap in the toolbox. So one of which is KC7. It is immunosuppressive. So immunosuppression means, I think, a lot of different things to different people. Not all arms of the immune system are created equal. For immunosurveillance, 2 key cell types are crucial, and that's NK cells, natural killer cells, and cytotoxic T lymphocytes. And I think this is best exemplified by our success in a number of immunotherapies, which really highlight the role of T cells for tumor immunity. So we've been doing some work at Amgen to evaluate the effects of drugs on the ability of T cells to invoke an immune response against cancer. So if you go to the next slide, one of the assays that we've developed is this T cell killing assay, which can be done either in vitro or ex vivo from PBMCs drawn from animals exposed chronically, or we could do it acutely with human PBMCs, for instance. In this assay, PBMCs are cultured in the presence of target cancer cell to assess T cell mediated cancer cell killing. And this is really facilitated by this BiTE molecule. Unfortunately, the animation didn't come through, but the point is the BiTE molecule here, one arm attaches or has affinity for a tumor antigen on the cancer cell, and the other arm of the BiTE molecule has affinity for CD3, a molecule on T cells, and it effectively bridges or brings together that T cell in proximity to recognize the human tumor cell and kill it. And that can be measured simply by looking at cell death, or actually more conveniently by looking at a marker of T cell activation, CD107A. If you go to the next slide, it just sort of illustrates some data demonstrating the response to a positive control like dexamethasone, which causes a pretty clear dose-dependent decrease in T cell activation. So I think this is a useful assay that can be used in vitro, ex vivo, can easily be applied in vivo as well in the course of clinical trials or other clinical monitoring. So if you For the sake of time, I'm going to skip the next example. So if you go to the next slide, I was going to focus on genomic instability and some of the approaches we're looking at on next-gen sequencing. But I'll leave you in suspense on that and just kind of go to slide probably 21, or 22 rather, just to kind of wrap up and leave a little bit of time for questioning. Okay, 23, sorry. More slides than I anticipated. So just to wrap up, so there's clearly still a lot of research to be done. I think establishing a list of assays is really just the starting point. I think depending on what's being evaluated and how the data is going to be used, a different set of assays might be appropriate. But I think there's still a lot of consensus to be built around what the right endpoints are, and of course, assays and associated protocols. Once that is in place, I think we can start more systematically evaluating the specificity of the assays and the KCs and the degree to which they are relevant for a meaningful cancer risk assessment. I think a key component of this is in vivo biomarkers, and I think that's probably where the largest gap is. But I think it's going to be crucial in order to understand the degree to which effects in vitro can translate in vivo. And if possible, start measuring this in humans as well. So I'll leave you on my last slide. It's really just an acknowledgement to the working group that I've been involved in, again, led by Martin Smith and others. So I'll just give a call out to them, and we're still actively putting that paper together to outline this list of assays. So I'll stop there and leave a few minutes for Q&A.
Thanks very much, Mark. So again, if folks have questions, they can put them into the chat window. We've had a couple of questions come through already. From Jay Goodman at Michigan State University, we have it appears that employment of the key characteristics of carcinogens concept or hazard identification is premature at this time in light of the fact that there is not anything approaching general agreement regarding what test systems might be appropriate for each characteristic. For example, there are numerous ways to evaluate oxidative stress. Which are best? What if one gets conflicting results? What model systems should be used? What are the criteria for dose selection, especially the highest doses? And the follow-up on that is whether Mark agrees that the employment of the key characteristics of carcinogens concept for hazard identification is premature at this time based on the lack of general agreement regarding which test systems might be appropriate for each characteristic.
Generally, yes, and that was kind of the point I was trying to make on my last slide. This is really just a starting point to start evaluating what are the right assays, protocols, the validation, the relevance. So generally, yes. I will say, though, that many of these endpoints, these KCs, are already evaluated in the course of the toxicology studies we're already doing, such as KC number 2 is genotoxic. Of course, we're already measuring this for pharmaceuticals. Histopathological results are informative for cancer risk, and we're already applying that to begin assessing pharmaceuticals. But of course, we're still employing the 2-year rat bioassay in the meantime. So of course, it's not a replacement at this point. But you're right. I mean, there's still a lot of consensus building to be had.
Okay, our next question is, I can see where these may be helpful in identifying drug targets, but where does duration and dose come into the hypothesis of the KCs? Yeah, and that's the tricky part with extrapolating results in vitro to in vivo. Now, for pharmaceuticals, obviously we're still doing chronic toxicology studies, and so being able to measure the KCs in the course of the chronic toxicology studies we're already doing is going to be extremely valuable, since you can imagine some false negatives in vitro. But I think that the mechanisms of action, which should be sort of acute endpoints, if appropriately designed, could be evaluated in vitro. So I think we're going to have to be able to enable an evaluation of these KCs in vivo to really plug all the gaps. Okay, next question. Carcinogenesis has been considered a multi-stage process. Is there consideration of how in vitro assays should be ordered, i.e. how to choose a test system that models the steps that precede a specific KC effect?
It's a good question and not something the working group has tackled. And I mean, that's a big question. I just don't think there's an answer at this point. You know, we're sort of at baby steps right now.
Okay, and then our final question before we run out of time is a logistical question. Where can we download the slides from both this and last week's pre-workshop webinars? They will be available on the workshop website, on the NIEHS Converging on Cancer workshop website, and we will send out an email to all of the registered participants when those slides are available. And then the last thing I want to remind folks about is that we have sent out the breakout group questions. We will resend those later today, and we are looking for feedback from those of you who are not able to attend the workshop in person next week and will therefore not be a part of the breakout group discussions on the day. We would still like your input on those breakout group questions, so please send us your feedback or look out for that email later today and get us your thoughts ideally before Thursday. So thanks so much to both of our presenters this week and to Cynthia and Glenn for excellent talks last week as well. And thanks to all of you for tuning in. And I look forward to seeing some of you next week and to getting feedback from those of you who will just be online.